Hello and welcome to episode 7 of CCLG's Research Talks for Parents and Public. I'm Ellie Wilkinson and my job is to share research with our supporters. So starting with some housekeeping, we'll be holding a Q&A session at the end of the talk. So please do submit your questions whenever you think of one using the Q&A button that is at the bottom of your screen. If you would like to use the chat function, make sure you've set the to drop down to everyone or people may not see your message. The webinar is being recorded and will be available publicly on our YouTube channel. We will send out the link when it's ready to watch. So this month's talk is called Why Can't We Cure Brain Tumours and is being led by Dr Stuart Smith. Stuart is a brain surgeon and Associate Professor of Neurosurgery working in the Children's Brain Tumour Research Centre at Nottingham University Hospitals. He researches how genetics affect the way brain tumours develop and how to use this knowledge to change cancer's behaviour. So without further ado, I'm going to hand over to Stuart.
Great, thank you. And I'll just start sharing my screen. Hopefully everyone can see that. And you can hear me okay? Great. Yeah, so thank you very much for the kind introduction. My name is Stuart. I'm an academic neurosurgeon at the University of Nottingham, working in the Children's Brain Tumour Research Centre and looking at brain tumours. My particular interest is in gliomas, high and low grade gliomas in children and adults. We run research programmes in both. But today we'll be talking mainly about paediatric brain tumours. We've been very fortunate to receive funding from the Children's Cancer and Leukemia Group and the Little Princess Trust to undertake some of this work. And it's a real pleasure to be here to talk to you all about brain tumours. So the title of the talk was actually from a patient who asked me, quite frankly, I think in clinic, why can't you cure my tumour, and why can't you do better? And I think it's a pretty profound question, and it really is sort of runs to the heart of everything we do. Why aren't we better at treating brain tumours, and why can't we make things better for more people? And I think putting this talk together actually for me has been a very useful exercise in sort of really fundamentally questioning, why we're going wrong in some cases, and why we can't do things better, and why our research hasn't improved things for more people. Lines aren't moving on. There we go. That's it. So, as I'm sure most people are aware, the brain and the spinal cord are what make up our central nervous system, which is the core of our nervous system, and that's obviously the part of our humans that control the rest of the body and make us who we are. Our thoughts, our feelings, our emotions, control of our actions all come from our brain via the spine and the other nerves in the body. But the central nervous system really is the operating system for what makes us humans. The brain is an incredible organ. It's endlessly fascinating. We're still only really scratching the surface of how our brains work, and neuroscience and the study of the brain is a huge field in its own right. But we know that there are billions of cells, billions of neurons in the nervous system, and there's more connections between all those cells than there are stars in the galaxy. It's an enormously complicated arrangement, much more complicated than any known computer, and does incredible things that no computer could do. But unfortunately, it does go wrong. And one of the things that can go wrong is the development of tumours. So all of those cells each one is in its own right an individual living package. When tumours arise in any part of the body, when those cells' instructions go wrong and the cells start to proliferate in an uncontrolled fashion. Now, normal cell proliferation is a normal part of most organisms and most organs within the body. But in the brain, particularly when that proliferation gets out of control, that can set up an uncontrolled growth of the tissue and leads to a tumour or cancer. And that can be quite an aggressive, fast-growing process. One of the things that makes brain tumours particularly complicated is that there's lots of different cell types within our brain, and each cell type potentially can give rise to different sorts of tumours. So we're not just one brain cell. There's neurons, which are obviously the sort of maybe the most widely known. They're the ones that help us think and do the sort of the actual work of the brain in many cases. And there's the black and white picture is an electron microscopy picture of an actual neuron. And you can see there's all those fine threads coming out from the main cell body. And in the brain, they all make contacts with lots of other threads from other cells. And that's what gives the brain its complexity and its ability to process information and do all the amazing things that our brains do for us. But neurons don't exist on their own. There are lots of support cells, which we call glia or glial cells. And even they come in different types. So astrocytes, oligodendrocytes, microglia, all do slightly different things. They support the cells, they wrap the neurons in an insulating covering to help the electricity flow along the neurons. And they nourish the neurons and support them. And actually, tumours of neurons, or tumours of mature neurons, are actually very rare. But tumours of the precursor cells and the glial cells are much more common. And we'll talk a bit more about some of the more common tumour types a bit later on. Ependymal cells are the cells that line the fluid spaces within the brain. We all have fluid spaces, as in the bottom right picture, those purple sort of twisty bit in the middle is the fluid spaces that we all have, and they're lined with something called ependymal tissue. And the ependymal cells are also a different type of cell. And again, they can give rise to tumours, particularly in childhood. Choroid plexus is the material within the ventricles that produces the fluid that bathes our brains. And again, you can have tumours of that. The meninges is the membrane that covers the outside of the brain. And even that can give rise to tumours. You can have blood vessels and immune cells. And all of these things, if they go wrong, if those cells lose control, they can give rise to tumours. So most of the cells in our nervous system grow very slowly, and probably in adults hardly at all, except in very special circumstances. In the childhood, there is still development. New neurons are forming, particularly in early childhood. There's a lot more growth and development, and that may be one of the reasons that brain tumours are relatively more common in children compared to adults. That brain tumours are one of the most common tumour types in children. And unfortunately cause a great proportion of the disease burden of tumours and cancer in childhood. But in all of us, even in adulthood, there's a sort of special population of cells within the nervous system called stem cells that act as a kind of reservoir. And particularly, we know that in some circumstances, particularly if there's damage to the brain, those cells can activate and start to divide more rapidly. And they're particularly active in children, we think. And these cells have the potential to become either neurons or glial cells and have that stem-like potential to go on and develop into numerous cell types. And to some extent, we think that tumours are hijacking that process of growth or damage response or development. And that process is the one that's going awry in cancers and giving these cells the capacity to divide out of control and become tumours. How does that process go wrong? Well, ultimately, it comes down to the DNA that all of us have in all of the cells of our body. That's what provides the instructions for those cells how to behave. And ultimately, that DNA in tumour cells is damaged. So programming, those instructions go wrong. And the cells escape the normal control mechanisms and divide out of control. However, how that damage occurs, we don't know in most cases. There are some very rare syndromes that again can give rise to childhood tumours, things like neurofibromatosis, tuberous sclerosis, or Li-Fraumeni syndrome, which some people on the call may be more familiar with, but those clearly predispose people to brain tumours. But the majority of even the children with brain tumours don't have one of those syndromes. We know that radiation can cause tumours, and in former times when children were given radiation, even for things like ringworm on the scalp, we know that that obviously predisposed them to tumours. Thankfully, it's been a long time since that's been done, and most children aren't exposed to radiation except maybe in the course of treatment for things like leukemia. There are carcinogens in the environment around us. For example, obviously well known in things like cigarette smoke, but also other carcinogens, chemicals in the environment that can damage the DNA and cause tumours. Interestingly, most of those don't actually get to the brain because of something called the blood-brain barrier, which is a biological barrier between our bloodstream and the brain, and only allows things like oxygen and sugar generally to cross over. We'll come to that more because that's really important as a source of problems in the treatment of brain tumours. One thing I would say is we're commonly asked, is it my mobile phone, is it Wi-Fi, is it broadband that's caused tumours? This has been looked at a lot, and it's really pretty well established now that these things don't cause tumours. I hope people can be reassured that it's not your mobile phone that's causing people, children or adults to get brain tumours. I'll talk briefly about some of the different more common types of tumour and some of the special problems that those individual tumours might have in treatment. But each tumour is different, has different features, and clearly it's unlikely there's going to be one treatment that covers every single type of tumour. Probably the most common type of malignant aggressive brain tumour in children is something called medulloblastoma. That's generally found in the bottom part of the brain, as in the diagram on the screen, and these tumours start in that area of the brain. A scan of a patient of mine unfortunately suffered from the medulloblastoma. It's the bright white area in the sort of triangular bit at the bottom of the screen on the top image, and the sort of larger grey area between the white ribbon-like structure and the leaf-like structure on the other side. Medulloblastomas arise from different types of stem cells, we think, in different parts of that area, the lower part of the brain. But even then, we can subdivide medulloblastoma, and one of the research has shown us is that most of these tumour types can be subdivided into different types, depending on the exact damage to the DNA that's driving the tumour forward. So medulloblastomas, as an example, can be divided into 4 variants. The names for them there, and these are named after the genes or the genetic pathways or the DNA that is damaged or different in that particular variant. So one of them is wingless, sonic hedgehog, which was named after the gene, which was named after the computer game character. And I think they ran out of names at that point because the other 2 were group 3 and group 4, which are probably less imaginative, but are the other 2 main types of medulloblastomas. So one of the issues that medulloblastoma and certain other aggressive brain tumours can do is they can spread through or even out of the nervous system. And medulloblastoma is particularly prone to doing that. And we know now that the different types of medulloblastoma have different prognoses and probably will need different treatments. So the wingless or Wnt tumours, actually the prognosis even with current treatment isn't perfect, but it's very good. Over 95% of children with that particular type of medulloblastoma will now survive and hopefully have long-term survival from the tumour. But unfortunately, for other children with other groups, such as group 3 medulloblastoma, the prognosis remains much worse and unfortunately can still be life-threatening. Other types of common aggressive tumours in childhood are high-grade gliomas. These are tumours of those support cells, the glial cells that surround the neurons. And glioblastoma is the most common malignant brain tumour in adults, and that can also occur in children. But children are particularly prone to certain other types of high-grade glioma, such as diffuse midline glioma that used to be called diffuse intrinsic pontine glioma, or DIPG. And that is driven by a particular damage to one of the parts of the DNA called the histones. It's H3K27M type of tumour is label means that tumour is driven by a particularly well-recognised issue with the histone part of the DNA. And that's particularly found in childhood tumours. It's reasonably rare in adults, but a lot of these diffuse midline gliomas in children are driven forward by that particular abnormality. And there are other types of high-grade glioma in children, and indeed the types of glioblastoma that adults get can also occur in children. Other types of common brain tumour in children are the pilocytic astrocytoma. Now, this is categorized in the histological classification that we use as a grade 1 tumour. And unfortunately, that's often referred to as a benign tumour. Now, I think most most children and families who are affected by this would say it's anything but a benign disease. It can be life-threatening, it can cause blindness, it can cause many very severe difficulties for patients. And I think, it's really best to try and avoid the terminology benign. And we would refer to this as a grade 1 or a low-grade glioma rather than a benign tumour. It's driven by another particular type, in many cases, of DNA damage called BRAF. The BRAF gene can be particularly prominent for these tumours. And it affects particular parts of the brain, such as the hypothalamus, which controls hormones and other daily cycles within the body. But it can also affect places like the optic nerve, so I say, giving rise to blindness, and also can affect the same parts of the brain that the medulloblastoma affects in the posterior fossa, that lower part of the brain. Finally, the other type I'd like to mention is the ependymoma, which is the tumours of that lining of the fluid spaces of the brain. And this is an overgrowth of those particular cells, which is different to the other types we've mentioned. So ependymoma is a tumour that, if we can remove all of it in an operation, can do very well and can be cured with complete surgical resection. But again, once the tumour's spread, or if it recurs after surgery, unfortunately, it becomes much more difficult to treat and much more problematic. There are many other sorts of brain tumours, and I'm sure there may well be people on this meeting who've suffered other sorts. And I apologise if I don't go into your type of tumour in detail, but there are many, probably up to 100 different types of brain tumour. They're all different, they're all special, they all have their own problems with the DNA instructions, and they all have their own treatment. So how do we treat people with brain tumours? So the sort of conventional ways of treating tumours are through surgery, as I've already mentioned. I'm a neurosurgeon, I operate on young adults and adults with brain tumours. We can give tumours radiation, which is like a high-powered version of X-rays, again, to damage the cells and try and kill them that way. We can give people chemotherapy, which is drugs, either tablets or into the bloodstream, to try and poison the tumour cells and eradicate them that way. In recent years, we've seen an increase in the use of targeted molecular agents, targeting those particular specific difficulties in the DNA that we mentioned briefly for some of the tumour types. So these are often drugs very much targeting a particular mutation or a particular abnormality in the cell. And probably this is the beginning of personalised medicine, where we can target treatments to individual tumours, hopefully with much less damage to the surrounding normal cells. Other approaches that are used either in other cancers or more experimental, but are sometimes used for brain tumours, are immune therapy, where the immune system itself is harnessed to target the cancer cells or the tumour cells. So my research, funded by CCLG and Little Princess Trust, have been looking at using electrical fields. And again, this is an approach which is used to some extent in adult brain tumours. We're hoping that maybe one day this will come to paediatric brain tumours. So before I go any further, I would like to say we can cure some brain tumours. So my title perhaps is a little misleading. So some ependymomas, meningiomas, pilocytic astrocytomas, for example, even some medulloblastomas can be very successfully treated with current approaches, but we can't treat everyone. And other subtypes of those tumours remain difficult to treat. So surgery is very good for some tumours, but we're limited by the extent of the brain function in the normal brain round about the tumour. Clearly, damaging normal brain or removing normal brain in an operation can cause catastrophic deficit, disability, and even be life-threatening. So surgery is limited. If we can get around the tumour and get some tumours out completely, surgery can be a very good treatment for some brain tumours. But other tumours, as I'll explain, surgery is never going to be the complete answer. Radiation, again, can be very effective, but there's a limit to how much the brain can tolerate, particularly the developing brain in childhood. and you've got more intrinsic cell turnover and cell division as the brain grows and develops, particularly in very young children. So if we give too much radiation, that would damage the normal brain and again cause disability or handicap. Chemotherapy, again, can treat some tumours very well, but obviously sometimes the drug doesn't get to where we need it to get to, and I'll talk about that a bit more. The novel or experimental approaches also have their drawbacks, and the targeted agents particularly only work if there's a particular target for that drug. If your tumour does not have that particular change or that particular mutation, then the drug won't work. But even then, resistance can develop, and the tumour can develop other ways of doing the same thing. The immune system is particularly inactive in the normal brain. It isn't well understood why, but the immune system doesn't work as well in the brain as it does in other parts of the body. So by stirring up the immune system with drugs or vaccines, sometimes even that doesn't achieve it. Electrical field treatment can use electrical fields put through the brain to suppress cell turnover and cell growth. Even that is not a cure, but it can, it has been shown in some adult brain tumours to extend survival. But we're making slow progress. Some tumours like diffuse midline glioma, some medulloblastomas, recurrent ependymomas, and glioblastoma are very difficult to treat. And we haven't made the same progress as we have for certain other tumours. Specifically, why are we struggling? The brain is very fragile. As I say, surgery, radiotherapy, chemotherapy can all damage the normal brain, and that limits how much active treatment you can give. The blood-brain barrier, tumour resistance to drugs, and tumour heterogeneity, which is the tumour evolving and turning into lots of little mini types of tumour, really limit how we can treat them. And the immunosuppressive environment limits the effect of immune therapy that's been very successful for other tumours. The blood-brain barrier is a major reason. So chemotherapy elsewhere in the body goes throughout the whole body and can treat cancer or tumours elsewhere in the body, and the drug gets to where you need it to be, which is right next to the tumour cell. Now, in our brains, there is this barrier between our bloodstream and the normal brain, which is there for good reason. It stops toxins in the blood getting into the normal brain and prevents damage to the precious brain cells. But unfortunately, it often stops chemotherapy getting into the brain to where it needs to be. If the chemotherapy can't get next to the cancer cell, then it won't work. So the blood-brain barrier is a major issue. You can only, if you give higher doses, you might be able to push a bit more into the brain, but then the systemic side effects of the drugs can become intolerable for the patient, and the drug may be at toxic levels. Heterogeneity is evolution in action. So tumours evolve both over time and also in space. The tumour here we see starts off as a relatively homogeneous population, but over time it becomes a collection of lots of different, slightly different tumour cells. And within that, some tumour cells will be more resistant to drugs or radiation than others. And that gives those cells an advantage. And the evolutionary process selects for those tumour cells and allows them to take over the tumour and allows the tumour to survive chemotherapy and radiotherapy and targeted therapies, for example. We know that tumours also evolve within space, and that in different areas of the same tumour, you can have slightly different populations of cells. So a targeted drug, for example, may work on one particular population of cells within one particular part of the tumour, but it won't treat the whole tumour necessarily. And so we have to be very careful when we're thinking about targeted therapy to make sure that we cover the whole tumour. As well as within individual patients, there's variation between patients. So glioblastomas in one child and the second child and the third child will be different. There will be differences between those tumours. Although they're all glioblastomas, they will all have their own slightly different DNA changes and their own slightly different makeup. Tumours resist drugs through various mechanisms. They can kind of go quiet for a while and become more like stem cells again, and that helps them resist drugs. They can develop what we call pumps in the membrane, where the tumour cells actually pump the drug back out of the cell, a bit like a water pump pumping excess water out of a basement. The tumour is actually able to pump chemotherapy out of the cell. Tumours can grow new blood vessels, they can suppress the immune environment, and they can change their nature and evolve, as I've described, to evade any treatment that we give them. Brain tumour cells, particularly things like medulloblastoma, glioblastoma, can invade into normal brain, following akin to these arrows here, and these tumour cells moving through different parts of the brain. And that makes them very difficult to treat because they can spread quite rapidly and quite readily beyond what we can see on the scans. And that makes it very difficult for us to target the tumour and know exactly where it is and the extent of the tumour. And it makes, because they're then intermingled with normal functional brain, that makes any treatment very difficult to administer without damaging normal brain cells. So what are the solutions? Where is the hope? Where is research leading us? And there is hope. And I'm going to tell you a story at the end of hope that I think will hold promise for many brain tumours. So diagnostics are improving. Our MR scans are getting better and better. We can look at blood flow in tumours, like the top left colourful image. We can look at chemicals in tumours now, like the bottom image with the sort of wiggly graph. Our genetic sequencing, even our routine genetic sequencing that we do on the NHS, is getting better and better and better. And we do things now that weren't even dreamt of 10 years ago. And we learn a lot more about every patient's tumour by doing that. Scanning can be linked to other things like better delivery of radiotherapy, but also better delivery of surgery. So this is a picture of our intraoperative MRI scanner at Queen's Medical Centre in Nottingham, which is used for childhood brain tumours and adult brain tumours. It was funded by by a huge collaborative effort. Universities, NHS, charities, Children with Cancer Charity, for example, they help fund this, and this is now routinely used and makes our surgery more accurate and better. Here's a picture of some surgery using a dye called 5-ALA that's also now going to be trialled in children. And that the pink areas that you hopefully see shortly on the scan, that's areas of tumour that are hidden away from that main resection cavity. And this dye, 5-ALA, lights up these areas under this blue light and allows the surgeon to see them much more carefully. So hopefully we're leaving less tumour behind, but preserving normal brain. We can deliver drugs in surgery beyond that blood-brain barrier, either through putting wafers in or catheters in or gels in. And this is beginning to come to the clinic more and more, so we can deliver drugs beyond the blood-brain barrier and make them more effective, allow chemotherapy agents to get to where they need to be. And our intraoperative analysis, even genetic analysis of tumours, is improving all the time to allow us to learn more and more and more about these tumours, personalise people's treatment, and to make surgery and the other treatments that follow on much more effective. Radiotherapy is improving. The two images at the top, the colourful areas, are areas where the radiotherapy is actually being delivered to the patient using two different techniques. And then you can see on the right-hand image, there's a lot more spread of that radiation into the normal brain compared to the left-hand image. So we can target radiotherapy much more effectively, get more radiotherapy into the tumour, spare the normal brain from toxic doses of radiotherapy. We can, for some tumours, use things called like proton beam therapy, which the picture at the bottom from Cancer Research UK. So the bottom left image, the blue beam, conventional radiotherapy goes sort of through the tumour and out the other side, whereas proton beam has the potential to stop much more abruptly within the tumour and spare the surrounding brain radiation. And that's great for certain tumours where we want to deliver very local radiotherapy. For some tumours, it's not appropriate, and some patients actually conventional radiotherapy may be better than proton beam. But for certain tumours, proton beam can deliver great quality radiotherapy with fewer side effects and damage the brain and other surrounding organs much less. There's a lot of research going on how we can give drugs to sensitize tumours to radiation, or alternatively protect the normal brain from radiation. And these are really exciting areas to allow us to maximise this useful radiotherapy. I think has quite a bad reputation sometimes, causing damage. And radiation sounds awful, but actually it can be a very useful treatment. And if we can make it more effective and protect the brain, I think it will still be useful. Chemotherapy, drugs companies finally are waking up to the fact that a lot of their drugs don't cross the blood-brain barrier. And ourselves and other groups around the UK are working very avidly with drugs companies to try and design drugs that get into the brain better and give better treatment. We can open the blood-brain barrier to allow drugs in through things like focused ultrasound. And again, there's research in the UK going on to look at that, how we can get drugs into the brain by using ultrasound to temporarily open that blood-brain barrier, allow the drugs to leach out into the brain, and then allow the barrier to close itself. There's new targets being developed from genetic work and other work. We're delineating these DNA abnormalities within cells, and that allows us to target those abnormalities much more accurately and spare normal tissue, the blunderbuss effect of the old chemotherapy. So some of those targets are listed there, but things like we can target the sonic hedgehog or SHH medulloblastomas, and inhibitors have been developed to specifically target that particular molecular abnormality. Immunotherapy is improving, vaccines are improving, checkpoint inhibitor drugs that stimulate the immune system and prevent tumours from suppressing the immune system have been really promising in other cancers and are being applied to brain tumours. And hopefully they can be made to work better in brain tumours. And other things like CAR T-cell therapy, which has shown great promise in leukemia, is now being looked at in brain tumours as well. Electrical fields have shown promise in adult glioblastoma, and we're hoping that over time that can be transferred and augmented for paediatric brain tumours. Finally, it's important to remember that supportive care rehabilitation is also an area where a lot of research is going on, and we can allow hopefully more children to benefit from, allow them to recover from any toxic side effects of their treatment. And neuroregeneration is clearly another key area for that. If we could allow normal neurons to regenerate and to repopulate the brain and to repair the damage that's done by treatment, that could have an immensely beneficial effect. So what's the future? Clearly, we need to get better treatments. There's no doubt, everyone agrees our current treatments for many tumours are inadequate, and we need to do better. And there's huge amounts of research going on to enable that. I think preventing tumours from evading treatment is key. If we can stop tumours from evolving resistance, keep them as they are, I think that would have a huge benefit. We must remember with any treatment, the blood-brain barrier. It's a key barrier of why treatments that work for other cancers don't work for brain tumours. And I think the whole community and drugs companies need to be aware of that and think about that carefully. But there is hope. So for a type of tumour in adults called adult low-grade glioma that's characterised by something called an IDH mutation, a specific drug has been trialled, and the results have been published in the last few weeks, showing that, as you can see, the survival, the progression-free survival in the group that received the drug was much, much better than the group that were treated with conventional methods. And this is a targeted specific inhibitor of this mutation, and really has opened the door, I think, in gliomas in adults, or low-grade gliomas in adults, to hopefully a much brighter future. And this is again a tumour for which there's been very little progress over the last 10, 20 years. But this trial, the Indigo trial, and this drug, I think really do show us a bright way forward where science and research can help improve things for children and all patients with brain tumours. So I'll finish there, and I'd like to acknowledge firstly the patients, families and supporters that have helped support our research and research in many other universities and units around the UK. Our funders, the people that make all this research possible, particularly the UK CCLG and the Little Princess Trust, who funded our research, but lots of other organisations that contributed to our research and patient support around the country in all sorts of ways. Thank you very much, and I'm happy to take part in a discussion or any questions.
Okay, so firstly, thank you very much. That's really interesting, especially going through all the different types of tumours so many. I know that's barely even scratched the surface of all the different possibilities. So we have a couple of questions. So the first one is a little bit of a, give me a second. So we've had a question from a lady. Her daughter died from glioblastoma, aged 2 years old. So she had glioblastoma of the liver from a liver transplant at 9 months old. And that liver came from a glioblastoma patient. Apparently the risk was apparently very low, but she wants to know whether you think it's a risk that should be taken when they say that it shouldn't cross the blood-brain barrier, but in her case it did twice.
Yeah, I mean, that's clearly a tragic, awful event. I think, you know, it's correct that in general glioblastoma it's very rare. It's not unheard of. We treat, I think, maybe about 200 patients a year in the East Midlands with glioblastoma, and I think we have maybe one case every 2 or 3 years where the glioblastoma spreads beyond the blood-brain barrier into the rest of the body. So although it's very rare, much less than 1%, it's not unheard of. I think the more general question about, you know, at the moment, the national policy is that patients with brain tumours such as glioblastoma can donate their organs. Because of the very, very low risk of the tumour spreading to the liver, for example. I think it's a very difficult question. That clearly, from the other side of the coin, there is a desperate shortage of transplant organs, livers, kidneys, hearts, lungs, and you know, if we're too restrictive in who we accept donated organs from, then that would worsen the shortage, and people, as I'm sure you're well aware, die on the transplant list waiting for liver transplants, for example. So I think it's a very, very difficult question, and clearly any recipient of a transplant will have immunosuppressive drugs to suppress the immune system, and again that can allow any cancer or tumour such as glioblastoma in the transplanted organ to grow more quickly than it would otherwise. So I think it is unfortunately a very complex, difficult question, and clearly, you know, what happened to your family, your daughter, your granddaughter, is an awful, tragic event. You know, clearly better ways of screening for tumours and better treatments for brain tumours would hopefully lessen the chances of something awful like that happening in the future.
Okay, so another question. So what is the overall survival rate for ependymoma with surgery and chemo, as children under 3 cannot have radiotherapy? Is the overall survival better in children who can also get the radiotherapy?
So I'm afraid I'm not an expert in ependymoma. There's certainly my colleagues in the Children's Brain Tumour Research Centre who are. Certainly, we know that the people who do best with ependymoma, the children who do best, are the ones in which we can remove all of it surgically. So if we can achieve a complete surgical resection in children or adults with ependymoma, then those people do very, very much better and have can be cured. So if we can achieve genuine complete resection, then that can be curative. There's a lot of studies going on led by Professor Grundy, Dr Richmond, and other people in Nottingham who look at ependymoma, particularly looking at the value of radiotherapy, chemotherapy. Obviously, as I say, the developing brain, particularly in very young children under the age of 3, is known to be particularly sensitive to radiotherapy, and the damage and the side effects of radiotherapy are thought to be very much worse in very young children compared to older children and adults. So clearly they are, there is a lot of interest in avoiding radiotherapy or sparing those children from radiotherapy, either by just using surgery or surgery and a form of chemotherapy. And there are a lot of trials going on, as I say, led by colleagues in Nottingham and other units, looking at ways of sparing radiotherapy for very young children, but maintaining survival and hopefully curing as many children as possible. But certainly once ependymoma starts to come back, and particularly after surgery and radiotherapy, then it can be very difficult to treat, and certainly it's an area where we need to do better, and we need, you know, more research, more trials, and newer drugs and approaches to make things better for those children.
Okay, so a question that sort of follows on from this. So adult brain tumour research seems to be further advanced than children. Why do you think this is, particularly for tumours that are more commonly seen in children and young people?
I think there is, in some ways, in some tumours, I think research for children's brain tumours is actually probably more advanced than adults. I think particularly for medulloblastoma, I think the treatment for medulloblastoma, which is predominantly a tumour of childhood, is much more scientific and much more targeted and much better than the treatment for many adult brain tumours. I think there's probably more research into adult brain tumours, particularly because glioblastoma is so common in adults. But really, I think we're only just beginning to make progress for adult brain tumours. And I think there needs to be more research for all brain tumours of all ages, I guess is the short answer to that question. Brain tumours, unfortunately, are still the most common cancer-related cause of death for children and adults under the age of 40. So both children and young adults suffer disproportionately from brain tumours. And that's wrong. That's a dreadful situation. These are all people with their lives ahead of them, and who we, in some cases, cannot offer any really effective treatment to. So the amount of research funding generally on brain tumours is disproportionately low for children and adults. And I think the charities clearly help make up that shortfall, but there really needs to be a sort of a national initiative and a national conversation about why that funding is so low, and why we're not making the progress that we should.
Okay, so next question. I think there is sort of a perception that drug companies aren't interested in developing drugs for things that aren't going to be profitable, like if children's tumours are comparatively rare. And you said you work with drug companies. I was just wondering sort of what the response was, whether you were finding that to be an issue.
I think it's certainly an issue. Undoubtedly, from a capitalist or commercial point of view, clearly the market for a new prostate cancer drug or breast cancer drug is going to be, many tens of thousands of people a year just in the UK. Clearly, even the most common adult brain tumour, glioblastoma, affects maybe 3,000, 3,500 people a year in the UK. Most paediatric brain tumours are affecting clearly much, much smaller numbers than that. And as I say, we unfortunately now realise that there probably isn't going to be a one-size-fits-all solution, and that the individual tumours are going to have individual treatments and individual solutions. Now, there are schemes, there are incentives, I think, for drugs companies to work on these rarer tumours, and clearly charity funding, academic funding helps to make up that gap. And if a lot of the work can be done by those sorts of sectors, then it de-risks things for industry. They're not, although there is clearly a commercial imperative, I think drugs companies are humans. They are parents. They are the people like the rest of us, and they do have sympathy for the plight of people with brain tumours, children with brain tumours, and there is a certain amount of willingness to try and help. And my conversations, and I'm sure other people's conversations with drug companies have generally been positive. But brain tumours are particularly difficult to treat, and designing drugs to get across the blood-brain barrier is difficult. It's not. Maybe I may apologise if I made it sound too simple. It's not just a question of wanting to do it. You need very skilled chemists, very skilled pharmacists, pharmaceutical workers to re-engineer chemically drugs that will do that. And that is a big problem. But people are working on it, and I think companies are one of the kind of side effects, I guess, of adult brain or adult cancer research is now things like melanoma can be treated often very well in the rest of the body, and melanoma patients in adults, particularly, the relapse often happens in the brain because the drugs that they're using to treat elsewhere don't get in the brain. So actually, companies now are starting to look at it and develop the techniques to get drugs into the brain, and hopefully one of the kind of byproducts of that will be designing better drugs for specifically for brain tumours.
it sounds more promising than it is perhaps often portrayed.
I think there is hope, and I think, as I said at the end, the Indigo trial and the IDH inhibitor story is a really good example of learning more about a brain tumour and putting that knowledge into action and developing a drug that really now seems to be very promising. We're very hopeful we'll be available to patients with that particular type of tumour in the UK, hopefully fairly soon.
Yep.
But I have to stress it is a targeted agent that is only applicable to that particular type of tumour, which unfortunately is very rare in children. It's a type of tumour that's most prevalent in I suppose the older teenage group and people in their 20s and 30s, so young adults, but it is a type of tumour that's not very common in children, and that drug is very specific for that type of tumour. So I think I tell the story not to sort of say, no, it's not going to work for children, but as hope, and show that research can lead to benefit and will change things, but it takes time. We've known about the IDH mutation for probably 10, 15 years, and it's only now that this drug is about to hopefully come to market and to fruition. So it does take time. It's a long time frame. It's too slow, and there may well be ways we can speed it up, but there is hope, and I think things will get better over the next 5, 10, 15 years for all types of tumour.
Okay, so we have a few more questions if you still have time.
Yeah.
Okay, so I've got questions that says, why is the future inhibitors for glioma only for adults?
Yeah, so it's, as I say, it's specifically targeting one of those DNA abnormalities. So it's targeting something called a mutation in the IDH genes. And that is very specific. It really only occurs in a particular type of tumour that's found in adults, and it's an adult low-grade glioma or adult diffuse astrocytoma or oligodendroglioma. But those tumours are really only found, they really only occur in people in their 20s and 30s. It's not generally a type of tumour that we see in childhood or younger childhood. And that's because it's very targeted and very specific. Hopefully it has much fewer side effects, and it's very effective against that tumour, but it won't target other tumour types. Now the hope is that these other mutations, these other abnormalities, the histone mutations, the sonic hedgehog mutations, they will also be amenable to specific targeted drugs that will work for that particular type. But they wouldn't work for adult low-grade glioma, for example. But it just so happens that this seems to be the first one that's really starting to work and show really, really great promise. But as I say, there's a lot of work going on for all of those different targets that I've mentioned, and we're very hopeful that over time, more and more targeted drugs, will be shown to work that are much more effective against the tumour, but much less toxic to the brain and nervous system.
Okay, so I think we have one last question. So somebody would like to know, how does Gamma Knife work? They said they've only heard of this, and is it used for brain tumours?
So Gamma Knife is a particular type of radiation treatment. So normal radiation treatment is generally given as a series of relatively small doses over a period of generally a few weeks, with the patient having a small dose repeatedly over maybe 25, 30 sessions, for example. The Gamma Knife is different in that it's one quite large single dose that's given very accurately and very targeted in a similar-ish sort of way to proton beam. It's very targeted. It targets an area. You can draw a circle around the area and just zap that area very, very strongly with radiation. It's quite widely available in the UK. Most neuroscience units now have that capacity. It's given slightly different names. Different companies make slightly different machines, and it's delivered in slightly different ways. So it can be called Gamma Knife or stereotactic radiosurgery or LINAC radiosurgery. They're all essentially the same technique, but given slightly differently. So it's very useful for certain types of brain tumour that don't invade into the brain. So things like meningiomas can be treated with that. Metastases, that secondary tumours from tumours elsewhere in the body, like a lung cancer or a kidney cancer that's spread to the brain can be treated very effectively with gamma knife. But it is only used for tumours that don't invade into the brain, because if you gave the brain that much radiation as one dose, you very severely damage the brain. So we have to very much restrict it to tumour tissue and only to tumour tissue. So unfortunately, for things like medulloblastoma or glioblastoma or diffuse midline glioma, where the tumour is intermingling with normal brain and invading into normal brain, it's not a suitable treatment for those in general. And its use is restricted to tumours that don't invade so much into the brain. But it's used very commonly in adults. It's not used so much for children, and that's purely because of the type of tumours that children get are different to those that adults get. It could be used for children, and I'm sure there are children in this country who've received it for the appropriate tumour types. But because of the different nature of childhood brain tumours, it's not generally widely used for those, but it is available for appropriate tumours if needed.
Okay, so I think that is all of our questions. So I will wrap up if that's okay. So thank you very much for everyone for joining us this evening, and also thank you to Stuart for your fantastic talk and many answers. This talk has been recorded and we will be sending it out over the next few days if you'd like to share it or watch again. You will also be receiving an email shortly to ask for your feedback on today's talk. This is so we can make sure our talk are the very best they can be, so please do take a couple of minutes to let us know what you thought. And thank you, and have a lovely evening, everyone.